Hello, this is Mr. Burfind, and today we're going to be doing a screencast based on the subject of macromolecules. You can see on the screen that we have some learning objectives. The first one is to recognize that cells are composed primarily of a few elements, such as carbon, hydrogen, oxygen, nitrogen, and phosphorus. And the second one is to compare and contrast the components, structures, and functions of each of the four macromolecules, and those four are carbohydrates, lipids, proteins, and nucleic acids. Alright, so here we are. You can see at the top that atoms are the building blocks of life. So when we look at all the different atoms that are present in those four major macromolecules we just talked about, you'll see that the primary elements are carbon, hydrogen, oxygen, nitrogen, and phosphorus. So what we're going to start out with is by just looking at the different four macromolecules and the structures of each of those, and we'll look for things that are consistent, so those main five different elements that are there. So we'll start out with carbohydrates. So carbohydrates, we have a basic monosaccharide, which is glucose, and we have a disaccharide, which is sucrose. And you can see in the glucose we have carbons, hydrogens, oxygens. All the reds are going to be oxygens, the blacks are going to be carbons, and those kind of grayish ones are going to be hydrogens. So you see we only have three of them right in here. And if you look at the disaccharide, it's the same thing. All these little points right there represent where there's a carbon. But look what's off of this one. There's an oxygen and a hydrogen. Carbon, hydrogen, oxygen. Carbon, hydrogen, oxygen, hydrogen. So the only components of carbohydrates are those three atoms. You can see for lipids, we have two different types. We have saturated and unsaturated. You can see that in our saturated molecule, here's a hydrogen and oxygen, otherwise carbons all across that backbone, and hydrogens coming off of them. Even unsaturated, only difference, we have a double bond right here. Otherwise it's all carbons, hydrogens, and some oxygens coming off there as well. So still just those three elements that we're looking at. When we get into proteins, we have amino acids, so this is an amino acid here, and this is amino acid here, and they are bonded together in this molecule here then. So this is the start of what we'd call a protein, and then we have a water given off in this reaction. But if we're focusing on just the primary elements, we have new ones, the purple, nitrogen, there, you can see even when the reaction comes together, there's the nitrogens, but otherwise, carbon, carbon, hydrogen, oxygen, 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 it's the same stuff. You put them together in a protein, and down here, reds are still oxygen, these kind of grayish ones are your carbons, and there's your hydrogens. So it's still just down to those three or four major molecules or atoms at this point. When we get to nucleic acids, now we're starting to see some nitrogens in here a little more often. There's some nitrogens, because we call this a nitrogen-containing base in a nucleic acid. And then we have our sugar, which would be the same as the sugars we'd see over here, because deoxyribonucleic acid is a sugar. And then now we have a phosphorus. This specifically is a phosphate group. So a PO4 with a -2 charge. That's a phosphate group. So a nucleotide is a nitrogen-containing base, a sugar, which is carbons, hydrogens, and oxygens, and a phosphate group. So now we've got all five of our different elements found in this nucleic acid. And you can see down here whether it's RNA or DNA, it doesn't change the whole basic structure. There's the sugar, the phosphate, and the base. There's the sugar, the phosphate, and the base. So the nucleotide unit is still together there. So the biggest thing for this whole screen though is notice the primary element, the reason why it's listed first, is carbon. Every one of these has carbons, and carbon is the backbone of all organic material. And the reason you can see it best here is that it's what we call tetravalent. Tetra means four, so 1, 2, 3, 4 bonds, and valent deals with those valence electrons. So what's doing the actual bonding to the hydrogen? This again is a covalent bond there, so this one can make 4 bonds. That makes it really special. It can be the center point of all the molecules that you'll see here. So now we're going to look at each of these 4 macromolecules in particular. So we'll start with carbohydrates. They're mainly used for energy. Think about sugars. We start out with monosaccharides. And so if we break this down, this is the monosaccharide is what we call the monomer. Notice how we have mono here, mono here. So what does mono mean? Mono means 1. So this is a monosaccharide, 1 sugar. Saccharide means sugar. So if you look at the monomer, it's a single repeating unit. So glucose is the most basic of the sugars. It has the carbons 1, 2, 3, 4, 5, 6 carbons. So its chemical equation is actually going to be C6H12O6. And so that's going to be the molecule of this glucose. The chemical formula is C6H12O6. So we talked about it being a monomer. So now let's put 2 together. And you can see that 2 has the prefix di. Di means 2. So this is a disaccharide. It's 2 sugars. Disaccharide. So you can see here that what we've done is we've taken this sugar molecule, this monomer, which is the glucose, and we've combined it chemically right through here with another one called a fructose. So together, they're called sucrose. Sucrose has 2 sugars attached to it. So di, 2, chemical bond, and now we have a disaccharide. All right, if we switch then to our final one, it's going to be called a polysaccharide. And poly means many. So I take this basic unit here, the monomer, and I'm going to link a bunch of them together. So that's going to create a polymer. Poly means many. So polymer is many repeating units. So I've taken this glucose one, and I've done the same kind of chemical reaction that we saw here, and now I've bonded them together here. There's a covalent bond, there's a covalent bond, there's a covalent bond, there's a covalent bond. This is what we call starch. It's a long-term storage area for energy. So carbohydrates store energy. They can be monosaccharides, 1 sugar, glucose, disaccharide, 2 sugars, sucrose, or polysaccharide, many sugars put together, called starch. All right, so that is our carbohydrates. We'll now switch to the lipids. And for the lipids, let me get rid of this stuff here. So for the lipids, they also are a storage of energy. So if we see our molecules for lipids, we have saturated molecules. Those are considered full. Think about if you're like after Thanksgiving dinner or after a big meal, you're saturated, you're full. The ground could be saturated with water. So when we look at this molecule here, how do we know that it's saturated? Every carbon is covalently bonded to 4 hydrogens. So you can see right in this spot here, we have this one is bonded right here. This one's here, this one's here, this one's here. That carbon is saturated. That's the most bonds it can possibly have. So a saturated fat has all single bonds. That's the big thing, single bonds. But if we look now at our second type of molecule, which we're going to call an unsaturated fat, unsaturated means that it is not full. So unsaturated, not full. So you can see the difference. All single bonds, straight chain. All single bonds, we got something different right there. There we have a double bond, and look what it did to the molecule. It went from straight to having a kink in it, which has an impact on the physical properties of this molecule. Unsaturated are liquids at room temperature. Saturated are solids at room temperature. It's because of this double bond right there. So could we fit more hydrogens in there? Yeah, we could put one right here, and we could put one right here. So that's unsaturated. There's room for more. So lipids are a storehouse of energy, just like we saw before for the carbohydrates. And then the next thing for this one is let's move ahead here. Uses. So now we have cell membranes. Cell membrane, you can see here is a whole bunch of carbons and hydrogens and carbons and hydrogens. There we have a kink, a double bond. This makes a hydrophobic end. There's a phosphate in here. This is a hydrophilic end. It likes the water. Now we have a cell membrane. So we're going to use those structures in order to have cell membranes. Alright, so that was our lipids. Next ones are proteins. So proteins complete most of the work in a cell. They're enzymes, they're collagen, they're all these other units that cause it for proteins. So an amino acid is the basic building block. So a monomer for a protein, again, a monomer is a single repeating unit. This is a monomer. So there are a bunch of different amino acids. And what we do to make a protein is we do a chemical reaction. We do a bond between these molecules. There's the water that's off. And now we see the bond here. We've taken this monomer and chemically bound it to that monomer. And now we have a chain of amino acids that we call a protein. So a protein is a polymer that has many repeating units. So the chain is extended here. And all these reds are the oxygens. So that was a monomer there, that was a monomer there, that was a monomer there, through that chain reaction. So amino acids then, if we look at what happens, here's our chain of amino acids. They start reacting with each other and making hydrogen bonds with each other. They start spinning and twisting and folding upon themselves. They start looking like this big, huge, tangled mess here, but that has a specific structure and a specific function. So now this is what we call the quaternary structure. This is what our whole body is made up of. So enzymes have a specific function depending upon their the organization of their amino acids, and that's the basic repeating unit that you would see here. Proteins are extremely important for us. All right, our last macromolecule is called nucleic acids, and you can see that nucleic acids store hereditary information. And so we're talking the same terms here. A monomer is a single repeating unit. So for a nucleic acid, the nucleotide is the monomer. So the structure, again, is a base with nitrogen, a sugar, you can see with carbons, hydrogens, and oxygens, and a phosphate group, a PO4. So there's that phosphorus with some oxygens. You take this base unit, or monomer, and you link it together into a chain, and now we're looking at another polymer. So many repeating units, DNA and RNA, are polymers. Take this base here and link them together. So you can see sugar, phosphate, sugar, phosphate, sugar, phosphate. So it's these two things again and again and again. And what's hanging off, these two things, and what's hanging off is this base, A, G, U, A. That's the name for a base that they have. So that's RNA, and DNA, same thing. Sugar, phosphate, sugar, phosphate, sugar, phosphate. That's what we call the backbone. And then we have our base in the middle with hydrogen bonds holding them together. So you can see just this side there is our repeating, that's our polymer, that's our repeating unit. And this is our monomer, which is the nucleotide structure. So you can see that there are some similarities in all four of these macromolecules down here, with them being carbons, hydrogens, oxygens, nitrogen, and phosphorus, but also the idea of monomers, monosaccharide, and then polymers, so a polysaccharide, or in here the monomer of the protein is going to be amino acids, and the polymer is going to be the protein, or even here the monomer is a nucleotide, and the polymer is the DNA molecule.